afternoon. I'd like to extend my thanks for everyone here and the opportunity to speak to you this afternoon. A little bit about our experience at the British Columbia Cancer Agency using the OncoScan platform. I'd like to thank Laura for the introduction. I know I'm a little bit of a complicated individual. I move around a lot, so it is true I did a lot of my training in Toronto and then moved out to Vancouver. And then largely because of my wife, we decided to move to Brazil because she's Brazilian. So we've decided to move down to this place here that's very, in the southern end of Brazil, a city called Curitiba, a population of roughly 3 million people. And interestingly, also a very high percentage of Japanese people in Curitiba and in the south of Brazil. So let's talk a little bit about the cytogenetics toolbox. We have a lot of different technologies at our disposal to help us perform diagnostic procedures for patients in constitutional, prenatal, and cancer applications. Conventional cytogenetics, of course, is everybody is familiar with banded karyotyping, and there are, of course, a variety of different flavors of that: G, R, C banding, silver nitrate, or replication banding, depending on what your particular application you need to do is. Molecular cytogenetic, this is basically two of the techniques that we used a lot at the Cancer Agency in British Columbia, fluorescence in situ hybridization, because we tested a very large number of paraffin-embedded tissues on a routine basis. So we were doing a lot of paraffin-embedded fish and some multicolor karyotyping combined with conventional karyotyping. And then, of course, part of some of the newer tools that are available now, including chromosomal microarray and next-generation sequencing, are new tools that are moving into the clinical spectrum. And exactly how we incorporate all these tools is somewhat of a question. Several years ago, of course, this was pretty much what all cytogenetics labs did. You took a specimen and you prepared it. You dropped cells on slides and you stained it. You looked at it under the microscope, and you analyzed it on a computer. And with the advent of FISH and other recent technologies, I have a laser pointer here. You can see that all of a sudden cytogenetic labs have started to become more versatile. We've started to perform not only karyotyping but FISH and microarrays and multicolor karyotyping. So all of a sudden the laboratory has become a much more versatile place, and we need to have a lot more technology experience with different technologies. And what's interesting about that is, of course, that this new technology has really made a boon to translational research. We're discovering a lot of things at the clinical level that we weren't seeing before using the technologies that we were using routinely because of questions like resolution or the types of abnormalities that we were looking at just weren't present. So this has become a really interesting area where we've started to understand a lot more about the biology of the human genome. And so as things become more and more complex and we get more and more tools in the lab, of course, we have to think about other considerations always, of course, like what's the turnaround time, which is always an important consideration, especially in cancer cytogenetics. What's the cost, and are there any ethical considerations to doing high throughput screening? And so a lot of people see this as the ultimate future for molecular diagnostics, but it's still a fairly long way away, depending on how you want to define that. This was I don't know if you've all seen this device that was prototyped at the AGBT meeting in Florida a few years ago, and it's a prototype of a next generation sequencer that would plug in to the side of your computer like a USB drive, and you would simply drop a small sample inside, and it would provide massively parallel sequencing for you. My understanding is this was a disposable unit, so it was about $1,000 US was the idea, and then you would just throw it away. So I don't know if that's cost effective or not, but it's certainly a very interesting idea. And then, of course, you're generating a phenomenal amount of data, a phenomenal amount analysis, and you have to find a way, at least in the clinical side, somewhere to keep all that information. And that's one of the current big questions that we're dealing with now in the clinic in terms of how do we store all of this data that we're generating. But when we started this pilot project, actually it was a few years ago, we were more concerned about adding microarray technology to our regular routine clinical processing, because we quite often had problems with samples where we didn't have a sample that was growing, so we had a failed culture, or we couldn't get live tissue from a patient, so we needed to be able to make sure that we could actually perform some kind of testing with it. And of course, in the clinical world, we often receive a very small sample, depending on the type of material that you receive. So of course, we had to be able to take a sample that we'd already received for conventional cytogenetics and try to obtain a result, and then potentially move that through to a microarray process and also get a result, because we weren't always going to receive a second sample depending on the type of disease we had. And so this is just a little picture of where this work was done at the BC Cancer Agency in Vancouver, Canada. And just to give you a little bit of a background about Vancouver, it's in the province of British Columbia. And if we take a closer look, you can see this is the province of British Columbia, has a population of approximately 4.5 million, the entire province. And most of those people live right down here in the bottom corner. And so right down here in this area, probably within about a radius of 30 square kilometers, we have 5 cytogenetic labs performing all of the cytogenetic services for the province of British Columbia. So that's actually a fair number of laboratories in a very small area. And so the BC Cancer Agency was primarily involved with clinical testing in cancer. And so we tested for a large variety of hematologic malignancies, as well as solid tumors. And we were doing a lot of this testing by FISH panel, occasionally by karyotyping, but quite rarely. And again, a lot of conventional testing here in hematologic malignancies, except for the non-Hodgkin's lymphomas and CLL, where we were doing a lot of FISH. And so obviously we wanted to bring a microarray platform into the lab to help us do diagnostic testing in the cancer realm. And we were, of course, there's a variety of different technologies that we can look at. And so we wanted to test most of these technologies to see what the data would look like. So this is a standard comparative genomic hybridization array where you would take 2 samples, your target sample and your reference sample. You would label them with different colored dyes and then hybridize them both to the array. And then you read the differential gene expression, as you're all probably aware. The Affymetrix modality, of course, is using your target sample all labeled, all the fragments labeled with biotin and then streptavidin and phytoerythrin. And this, of course, gives you a single color hybridization, which then can be read off the chip and compared to a reference pool of samples. And so one of the big considerations that we had asked, we knew at the time, and this was before the official release of the OncoScan platform to the public, where you could actually run it in the lab, but we knew that AFI was developing this platform for use with formalin-fixed paraffin-embedded specimens, and we wanted to also test all the different microarray platforms and their applicability with samples that had been fixed in methanol acetic acid, because at the time we were doing this, I had phoned around to a bunch of different laboratories and asked how many labs had actually taken their cytogenetic pellets and hybridized them to array, and had they had any problems with that. And nobody had really done a lot of work with this, so we thought, well, we better give it a try and see if it works. And of course, depending on what kind of fixation method you use for your cells, whether you use phosphate buffered formalin or non-buffered formalin, alcohol formic acid, carnoise fixative, which is a standard cytogenetic fixative, and a bunch of other types of fixative, you can get a very variable quality in the amount of preservation of nucleic acids. And of course what happens is that all of this material that gets fixed gets degraded, the fragment sizes get smaller and smaller, and then at some point it becomes very difficult to use them in downstream assays, whether that be quantitative PCR, other kinds of PCR, or microarray experiments. And so some of my colleagues at the Cancer Agency did some more work on this. This is actually just a nice paper for your reference if you're at all interested, looking at quantity and quality of nucleic acids from paraffin blocks, looking at optimal fixation times and DNA extraction techniques. And they've looked at paraffin, at fixation in buffered formalin, and also the extraction using a molecular fixative, which is basically 90% methanol. And so if anybody's interested, this is a really nice paper for if you're planning on doing any work looking at these kinds of paraffin-embedded specimens. So the OncoScan platform is just a little bit different, in fact, than the standard Affymetrix microarray platform, in that it's designed using this probe, which is called a molecular inversion probe, or sometimes referred to as a padlock probe. And what it is, this circular probe has the genomic targeting sequence that you're interested in. Here, 20 base pairs on either side of a space, and that space targets a single nucleotide polymorphism. And then in addition to this region, there's a DNA cleavage site, 2 universal primers, and a tag sequence, and it's this tag sequence actually that hybridizes to the array, as opposed to the target sequence hybridizing to complementary DNA on the microarray itself, as other standard microarray platforms operate. So what happens is you get this hybridization, the ligation across this extension ligation across the SNP site, and once that's occurred, this padlock probe can be cleaved, and then it's opened up into this linear format, and then you can amplify all of the hybridized events using a set of universal primers. So the advantage here for degraded samples is that you're amplifying selectively the material that you want to hybridize. So you're talking about less input DNA required, and you're not amplifying genomic DNA that's subject to biases that general global amplification of DNA would be associated with. And so the OncoScan platform, of course, has about 334,000 copy number markers, some of which are non-polymorphic, the rest of which have a SNP in them. And they're divided down into a bunch of functional categories. So you do get copy number information, you do get allele information or SNP information from this platform. And one of the other interesting additions to the platform was that you are getting information. All of these genes have, and what you can see is there's a bunch of different genes here, and these are genes that are routinely mutated in cancers. And the number to the right indicates the number of common mutations for that gene that are included on the array. So in addition to getting all of the information that you would get from a normal microarray, you're able to actually interrogate the mutational status of several genes all in one step. So in that sense, it's a very interesting product. And of course, it's important to remember that as a tool, a microarray is a part of a toolbox, as I said before, that has a lot of advantages and some limitations. And so these are the different types of genomic alterations that you can find in a sample. And the microarray is actually quite good at detecting a lot of these, but has some limitations in terms of, for example, detecting balanced reciprocal translocations, and in terms of interrogating, well, if you're not using a SNP array, of course, it becomes very difficult to sort things out like loss of heterozygosity. And so again, it's important again to look at the kinds of testing that you want to do in the clinic and decide what the appropriate tool is for that. So to talk about the pilot project that we did, we decided that we would test a bunch of different array platforms side by side and look at the data that they produced. And in order to do that analysis, we used a software package, Nexus Biodiscovery 6. And this is actually the software, the new version, version 7, that's being distributed with the OncoScan should you decide to use it. And so anyways, it's a very nice software for microarray analysis. The reason that we chose Nexus, and it wasn't officially distributed with OncoScan at the time, but the reason that we decided to do all of the analysis in Nexus was because of its ability to load microarray data from any microarray platform, and it was able then to do segmentation for all of the different samples from the different platforms using the same algorithm, and it gave us a side-by-side comparison and quality metrics using the same type of graphical interface. So you weren't trying to interpret a bunch of different images from a bunch of different software packages. And so the quality metric that the biodiscovery software produces gives you an idea of what your sample quality is, and basically in the guidelines for the software, it's somewhere at around 0.22, which is this red dotted line here. They say under this value is a good quality specimen. And so we had taken several fresh specimens where a bone marrow sample had come into the laboratory, and we had extracted DNA from those immediately. And then we had several specimens that we had sent through for cytogenetic processing, so they had been cultured and then extracted and harvested with methanol acetic acid, which is what MAA stands for. And so, of course, anytime you subject cells to this kind of fixation and processing, you can see here what we expected is that there was some loss in quality in terms of the quality metrics. However, when we looked at the actual data itself, the data was quite good from the OncoScan platform. This is a standard copy number plot, and the speakers that follow me today are going to talk about these plots in more detail and give you a more description. But just to basically describe this, this is a copy number plot where you would see deviations above this line here as a gain and losses as a deviation below the line. And this is the allele plot where you can see whether or not you have a standard Mendelian relationship from the SNP probes. Each little dot represents a probe, and the signal, whether you have, for example, an AA, an AB, or a BB SNP profile. And then in places where that particular pattern is deviated, you can see that the signal pattern looks a little bit different. So if we look at that area zoomed up, you can see that here we don't have any evidence of a change in copy number, but we do have loss of heterozygosity here. So this is a copy-neutral loss of heterozygosity event on chromosome 9p, which contains the CDK2NA gene, or P16, which is a gene that's known to be mutated in many cancers. So this is a very interesting finding and something that we would follow up on clinically. This is, of course, what you never want to see, and this is a plot from one of the poor quality specimens that we had processed. So I just wanted to show you very quickly just a couple of the actual cases that we ran. And if you're familiar at all with myelodysplastic syndromes or acute myeloid leukemia, you'll probably recognize a lot of the chromosome abnormalities here. And these are fairly complex cases in terms of metaphase cytogenetics, and you can see here loss of a chromosome 3, deletion of chromosome 5q. We have trisomy for chromosome 8. We have a small abnormality on the P arm of chromosome 11. When we weren't really sure what it was very difficult to tell because it was relatively small. A deletion of chromosome 17p. We're also missing a chromosome 20 here. And we had these two little ring chromosomes that were interesting. We weren't really sure what they were. When we looked at OncoScan, we were very heartened to see that, of course, the OncoScan platform very accurately represented what we saw by metaphase cytogenetics with the 3, the del5q, the trisomy 8, the small deletion on chromosome 11, also a deletion on chromosome 17p, which we'd also detected by metaphase cytogenetics. And we knew we were missing a chromosome 20, but what this actually shows is that we're missing 20q, and we have an amplification of this area on 20p. So probably these ring chromosomes are some combination of chromosome 20s. So that was quite good. We were happy to see that. And in a similar case here, also somewhat complex with many of the same abnormalities, we saw a small deletion chromosome 3, deletion 5q, let's see, we had a plus 10 in this particular cell. We had a funny bottom of chromosome 11 here, which we called an AD11Q14, minus 12, del14Q24, minus 15, minus 16, and then a couple of marker chromosomes down here in this composite karyotype. And so there's a lot of things going on, and when we looked more closely using the OncoScan again, we saw what we expected to see here with the deletion on 3, 5Q. And then when we looked here, and let's not forget the additional chromosome X. And then if we look at these abnormalities, we didn't see the plus 10 on the array, but it was only in a few cells from the metaphase cytogenetics. And then when we look at this region here, which we reported as such in metaphase cytogenetics, we can see a much more complex pattern of rearrangements, especially involving chromosome 14 and chromosome 15. So that we hadn't appreciated that there was this level of complexity with these two chromosomes. And of course, Laura had mentioned about chromothripsis before, and this is probably indicating that there was a fairly significant chromothripsis event in this patient, obviously indicating a poor prognosis. So one final case just very quickly to look at here. And this is an interesting case where we had a very unusual chromosome 10 here, missing most of the Q arm, and a derivative 119, so this chromosome 19 here with most of an extra Q arm from chromosome 1 added to the top of it, plus a very strange chromosome 21 with a bunch of additional material. And so again, when we looked at that using OncoScan, we could see that we were replicating all of these abnormalities very well. We could see that almost all of chromosome 10Q was missing, matching almost exactly what we saw in the metaphase cytogenetics with additional material of chromosome 10P, and both losses and gains on chromosome 19 and high amplification of chromosome 21, which probably contained a lot of the material from the AD21. And of course, it's very nice to see, using the OncoScan platform, that the copy number data and the allele profiles give you a confirmation of what you're looking at. So when you have losses, you can see that you don't have 3 allele tracks here. You don't have the 3 possibilities that you would in a diploid situation. And of course, when you gain copy number, you also have more tracks than you would in a standard diploid situation. So this gives nice confirmation about this information. And when we looked at some other platforms that also did a very nice job at detecting copy number changes across the genome, although some of them had some more difficulty with these kinds of fixed specimens when looking at the SNP profile. So of course, when you look at the SNP profile here from the AFI chip versus this, it's much cleaner on the OncoScan platform. So if we conclude what we learned from our very small pilot project, is that we looked at a bunch of systems that all detected copy number state well. There was a little bit more noise in the Agilent platform, maybe possibly due to a single reference sample that was used in that particular experiment. The SNP performance we saw on the AFI platform was really good in terms of both content and data quality. And so we looked at a couple of other options as well, including Oxford Gene Technology, which I didn't present today. But if you have any questions about that, I'd be happy to answer them. And so just very quickly to conclude, why would we want to use a microarray in cancer, and is it useful? And of course, there are always some difficulties in terms of analysis, and when you have mixed normal tumor cells in a population, and we can deal with those problems in using some techniques such as flow cytometry or microdissection or macrodissection, which if you have a relatively high percentage of tumor cells you can do. There's also genome instability in cancer, which is a problem. You're going to see that in your microarray. And then whether or not you have multiple clones or not, which can be related to a change that's present in all cells, or as you usually see in cancer, you can have many clones. And so the analysis of cancer microarray is of course growing, and there's more databases and a lot more information on different tumor types and their standard copy number profiles that are seen in cancer. And it's certainly for us to use a microarray for tumor types that frequently show aneuploidy and would require complex FISH that we were running to some extent in the lab, it was an excellent platform in addition for us to use. I'm going to skip that for today. And I'm just going to very, very briefly talk about some new challenges. So in Canada at the BC Cancer Agency, we of course had a very well-established clinical cytogenetic service. We were testing a great number of specimens per year, both in our laboratory and in the province as a whole. And in moving to Brazil, we've come up against a very different reality, of course. And genetic services and testing in Brazil is an area that is behind in many, many ways. And so what you can see here is a map from this recent paper I just showed the title of, showing where the location of genetic services are in the country. And of course, like a lot of countries, you can see the population of Brazil is concentrated here in the coastal areas, and this mirrors, of course, the location of much of the genetic services. And this is, many of you will undoubtedly have heard of the city of Sao Paulo, which is a very, very big city in Brazil. Somewhere on the order of 18 million people is right in here. And of course, the state of Sao Paulo as well has a great concentration of genetic services. And once you exit the state of Sao Paulo, you can see that there are very, very few genetic services. And including in our state here, Paraná, this is the city of Curitiba, where we have one center for genetic services. And so, of course, what this leads to is a problem where only about 25 to 30% of the estimated need in genetics is actually seen by a professional in the country. And so we're fighting an uphill battle, both in terms of clinical genetics referral, but in terms of laboratory services as well, because the two generally grow hand in hand together. So this is a picture of our new laboratory in Curitiba, Mantis Advanced Diagnostics. And we're hoping very soon to be providing services to our region and the state of Paraná. And with that, I would like to just say thank you and acknowledge some of the people that helped working on this project, and that if anybody has any questions about this experience, I would be happy to answer them.
.日本語でも結構ですんで。